Alright, so today we'll be talking about meiosis. And let's get started. Alright, like I said, we're going to be talking about meiosis today and the process that it goes through. Meiosis, mitosis is usually used for almost every cell division that your body needs, but specifically, meiosis has only one job. The one job that it is, or that you would say that is his job, is the production of gametes. Gametes are sex cells, either sperm cells or egg cells, and pretty much they're used, or their purpose is to make daughter cells with just half as many chromosomes as the starting cell. Meiosis goes through different phases. Meiosis goes through similar changes and has relatable strategies as mitosis when it comes to separating and organizing chromosomes. Meiosis has more to it. It goes into a lot more detail than mitosis. It still needs to separate sister chromatids like mitosis, but it also separates homologous. It's a two-step division process. Homologous is first in the process, and then it goes through the whole process of the sister chromatids second. Cell division happens twice in meiosis. One starting cell can produce 4 gametes, which is a prophase, metaphase, anaphase, and telophase. And we'll be going into detail about these phases throughout the PowerPoint. Meiosis 1, this is the first stage that it goes through. During meiosis prophase 1, meiosis begins to condense and also pair up. Each chromosome lines up with the homologue so that the two match up in corresponding positions. DNA is broken down after this because it has to be at the same spot of the homologous. And once the DNA is broken down, it reconnects in a crisscross pattern so that the homologous can exchange its DNA. And a protein structure called synaptomal complex. This holds together the homologous. In anaphase, homologous are pulled apart and moved to opposite sides of the cell. The sister chromatids of each chromosome do not come apart, and they just end up staying together. In telophase, the chromosomes arrive at opposite poles of the cell, and cytokinesis usually occurs around the same time as telophase. They form 2 haploid daughter cells whenever cytokinesis kicks in. In meiosis 2, this is the second stage that it goes through. Cells are able to move from meiosis 1 to meiosis 2 without having to copy DNA. It's actually a lot more simple. Like meiosis 2 is actually a lot more simple and has less detail and less productions and everything that they have to go through than meiosis 1. In prophase 2, chromosomes condense and the nuclear envelope breaks down if it is needed. Centrosomes move apart, the spindle forms between them, and the spindle actually begins to capture chromosomes. The 2 sister chromatids of each chromosome are captured by microtubes from opposite poles. In metaphase 2, the chromosomes line up individually along the metaphase plate. Then it goes on to anaphase 2. The sister chromatids separate and are pulled to the opposite sides of the cell, pretty much to the opposite poles. Finally, in telophase, nuclear membranes form around each set of chromosomes, and the cell chromosomes condense. Cytokinesis splits the chromosome sets into new cells, forming the final product, which is 4 haploid cells, in which each chromosome has 1 chromatid. The production of this is the sperm and egg cells. And this is the phases of meiosis. Thank you for watching, and I hope you have a good day.